Please, if you're interested, please let me know. So today's speaker is Alex Tsoi with Department of Dermatology, and I believe recently a faculty member for DCMB.
Right, thanks for the introduction. In today's talk, I'm going to share with you our efforts in identifying psoriasis-associated loci, as well as how we provide biological inference for those signals that we identified. So just want to give you some background about psoriasis. It is a autoimmune disease of skin. It has a prevalence of around 0.2 to 2.5%, depending on the population of origins. So, for example, in Caucasians, it has been estimated to be around 2.5% for prevalence, as estimated in studies in US, UK, or Germany. Over 100 million people with psoriasis worldwide, and in each year, over 150,000 new cases are identified in the US. So it is a complex genetic disease, has a heritability estimated to be around 60 to 90%. So in terms of dermatological changes, it has been observed in psoriatic skin, the immune cells will tend to infiltrate to the skin layers, okay, causing the increased proliferation rate of keratinocytes. Keratinocytes consist of over 90% of cells in the epidermis. This increased proliferation rate would cause the expansion of the epidermal layers. Okay, so the figure shows on the bottom here. On the left is the normal unimorphed skin. This is the psoriatic skin. As you see, the epidermal layer is actually expanded. Okay, this is because of the increased proliferation rate actually caused by the infiltration of the immune cells. So psoriasis could occur in different parts of the body, but mostly in the scalp, in the back, or in the hip. But it could happen in everywhere in the body. So it has significant impacts both in economics and public health. Every year, over billions of dollars have been spent to psoriasis-related healthcare. So in terms of health impact, besides that the psoriatic patients have lesional skins, they tend to have a higher risk of getting other diseases, such as other autoimmune diseases. And also they have a really significant psychological burden. So in fact, the stress or psychological disorders that they could get is actually compared to people that have more severe disease, such as cancer or cardiovascular disorders. So to understand the genomics of psoriasis, the Michigan psoriasis team aims to use different Hapmap technologies to identify the disease components, as well as to identify the disease mechanisms of psoriasis. So the Michigan psoriasis team consists of investigators from the dermatology, J.T. Elder, Johan Johnson, Andy Johnston, Regent Nell. Also investigators from the biostatistics, Gonzalo Abicases, Hyun Kang, William Wang. So we have been looking at two different scales, the genetic and transcriptomic aspects. For the genetic scale, we have been using different genotyping array and more recently target sequencing with three goals in mind. First one is to identify disease-associated loci. Then we also want to understand what's the impact for low allele frequency on the risk of psoriasis. Then we also have different customized genotyping array, such as we call it the follow-up array or Immunochip, aiming to do fine mapping to identify potential causal variants within disease-associated loci. So I'm going to concentrate my talk today in the genetic area, okay? But we also look at the transcriptomic scale. Some of the things that we have done, we've been using microarray and more recently RNA-seq to perform different co-expression analysis, identify different co-expressed modules that are different between psoriatic skin and normal skin. And we try to functionally characterize them by using functional annotations, as well as different cell type specific signatures. Okay, and more recently, in collaboration with Arul's group, we've been using RNA-seq to and develop a linked RNA identification and analysis pipeline, and we identify up to 1,000 novel linked RNA expressed in skin, and we provide functional inference for them and show that those linked RNAs have really specific cell type or tissue-specific expression or epigenetic profiles. So again, I'm going to focus my talk in the genetic scale. So before 2012, we and other colleagues have used different genetic association studies, to identify psoriasis-associated loci. The most notable one is the locus in the MHC, the HLA-C gene, which have the highest effect size for psoriasis risk. And altogether, we have identified over 20 psoriasis-associated loci in Caucasians. So all this are based on GWAS results. All these are really promising results, but they are actually relatively low-hanging fruit because they have a larger effect size, so they could relatively more easily to be identified. So in order to identify more disease-associated loci for psoriasis, which have more subtle effects, we need to have a cost-effective way to genotype people. And in addition, we have to be able to genotype that in large scale. So to address this, back in late 2010, we and other colleagues that investigating different other autoimmune diseases have jointly designed a genotyping platform called the Immunochip. This is we designed this so that we could have a cost-efficient genotyping platform for genotyping people for complex immune disease study. Other participating disease consortium include those investigators investigating, for example, multiple sclerosis, IBD, type 1 diabetes. So the goal for this Immunochip is actually to deeply interrogate around 190 loci, including, so in the Immunochip, there's around 190,000 genetic variants, okay? So among those 190 loci that we focus to study, including those loci that have achieved genome significance in previous GWAS in at least one autoimmune diseases. And also, we also include loci that show suggestive evidence of association in previous GWAS studies. And we also allow some investigators to submit some genetic variants within loci that they are interested to look at, okay? So we call it like a wildcard region. So by using Immunochip, we have genotype over 10, and we coordinate with a few other centers, and we have genotype over 10,000 individuals. And we formed this consortium called Psoriasis Association Genetics Extension page. Among this 11,000 genotype individuals, over 4,000 of those have psoriasis. And among those genotype individuals, around 1,700 of those are patients or controls in the. You have a question?
Does everybody have psoriasis in Newfoundland?
Sorry.
Sorry, I was just making a joke. Does everybody in Newfoundland have psoriasis?
Oh, no, they just do not provide the controls, right? Yep. So we have 1,700 people from here from UM. So because we focus our study to identify genetic associated to identify these associated loci in Caucasians, so we only try to genotype individuals that have that self-report to be Caucasians. So in here we are using genetic data, and we perform a principal component analysis with the Hapmap samples, and you can see that most of those samples from our different cohorts are actually overlapping with the European samples from Hapmap, which is good, but we still found quite a bit of a population outliers. in here, and then we just for subsequent downstream analysis, we just remove these population outliers. So then we perform genetic associations analysis by controlling the physical components using those as covariates, and we also use the individual center as indicator variable in the association. So one of the ways that we could use to assess whether there's any population stratification between cases and controls is to see is to plot that is the p-value of the association in the QQ plot, and then we could also calculate the lambda GC value if it's really close to 1, which means that there's not much population stratification between cases and controls. So we in this plot here, the y-axis is those the association from our result. The x is expected ones. We separate those markers from those that we separate markers into those markers that from region of previous associated loci, the red ones, and those that are outside the previous associated loci. Okay, so obviously the ones that in the region of associated regions actually are highly inflated, which we would expect. But for those outside the previously long psoriasis loci, we still seem inflated. Okay, so it seems that there may be some population stratification, but in fact we have to bear in mind that the Immunochip itself is actually biased towards genetic variants that in the immune-related region. So we could not just use all those markers outside those psoriasis loci to assess the population stratification issue. So we have to. So the way that we try to address this is actually that we look at GWAS results from we look at results from 3 previous GWAS studies. Okay. This is independent of our Immunochip. Okay, we perform a meta-analysis only based on 3 GWAS studies and identify markers that have p-value greater than 0.5. Okay, so we sim those as null markers, which are the markers that are not supposed to be associated with psoriasis. Okay, then we split the 2 different categories in my previous slide into these 4 different categories. So for those genetic variants that are within or outside long regions, we further split that into those have GWAS greater than or less than 0.5 p-value. Okay, and then we can see that now those genetic variants that are outside the known regions and with GWAS p-value greater than 0.5 is lie between the 95 confidence interval of what we would expect. Okay, so if the result suggests that the population stratification actually have been taken care of after we use those principal components as well as the individual center as covariates to control. Then the genetic control here, the p-value right is like it's a 1.2 now. It's like 1.05. So it is also another indicator showing that we do the population stratification is well controlled. So we did a small quality control step. So we go with the samples went down to 9,000. For our consortium, we also we then performed meta-analysis including the 3 published GWAS and then a independent Immunochip data set from our collaborators in UK. So altogether, the meta-analysis consists of over 30,000 genotype individuals, more than 10,000 of those cases. Okay, so we using Immunochip samples alone, we actually increased 2 times the number of sample size when compared to the combined GWAS samples. So this is the first. We performed the first meta-analysis in 2012. We confirmed 19 of the 21 known psoriasis loci which achieved phenotypic significance in our analysis. We further identified 15 novel loci achieving genome significance the first time. And then some of those interesting results, such highlighting the fact that skin actually participating in both innate and adaptive immunity. For example, for those loci that only appear in psoriasis but not in other autoimmune diseases, they have a high proportion of those include genes involved in innate immunity. And then more recently, we streamlined our code control and analysis pipeline by using refined genotype calling and imputation allowed us to increase 6 times the number of genetic variants when compared with the 2012 study. By doing this, we will find more low-for no-such genome-wide significance, and then altogether we double the number of G1 signals to 41 for Caucasians. So the first question that we would like to answer is that how we could better understand the underlying biological mechanism. So they are just like other high-throughput studies like microarray or RNA-seq. We approach this different ways. We could see if there's any functions that are associated with the genes from the disease-associated loci, or we could actually look at if there's any interactions between genes in different loci. Interactions could mean physical interactions such as protein-protein interaction. However, that we face different challenges when doing that. For example, there could be different gene candidates in one disease-associated locus. The regional association plot here show one psoriasis-associated locus. The best signal, so this is association negative log 10 p-value. The best signal here is in purple, and those red-colored genetic variants actually seem to be on the same haplotype of the best signals, and it's actually spanning different gene regions. And not to mention the fact that we now speculate a lot of those have roles in regulatory roles, so it could actually affecting genes like even further apart. Okay, so there could be different gene candidates, and also it's actually specific to this context that the Immunochip alone actually again a lot of the markers in the Immunochip is actually enriched in immune-associated region. So the background itself is actually immune enriched. So we want to have a better we find analysis to really review those psoriasis-specific associated immune functions, okay, or other related pathways. So just to give you an illustration of what I meant, so and the other existing approach that have addressed the two different things that I mentioned. So for doing functional enrichment analysis, we basically want to see if those genes in the associated intervals are enriched in certain gene function or pathway when compared with the background. Okay. At the same time, we do the interaction analysis. We want to see if those genes from the associated loci tend to be interact with each other directly, or they share the same labor in the biological lab work. And this we could access the statistical significance by randomly selected markers from different loci in the background, and then we see how many times they actually interact or share labels. Okay, but we propose to combine these different approaches so that we are able to distinguish pathways that only enrich in the genes with association within the association regions versus pathway not only enriched with genes in the association interval, but at the same time they're actually more coherent with each other and they're more directly interact. Okay, so this is just like another figure to show the rationale. So the rationale is that the genes participating in similar functions tend to cluster with each other in the biological lab work. This is a figure showing each node representing one gene, and this is a text mining derived gene lab work. So those different colors representing different functions, and you can see that those genes that participate in different functions kind of tend to like cluster with each other. I mean, this is well known. I just want to show you that that's the rationale for what we proposed. And at the same time, there could be different interactions between any two genes. There could be direct interaction. There could be shared labels. There could be more than one intermediate nodes here. There could be no interaction at all. And we also could deal with different types of biological lab works. Okay. So so we proposed to integrate those two ideas, and we developed this approach called MEAGA, minimum distance-based enrichment analysis For genetic association, again, the rationale is that genes from the disease-associated loci are closer, that in the disease-associated pathway are closer with each other in the actin. Okay, so the overview for this for MEAGA is actually shown here. So from the association analysis, we pick the best markers. Sorry, we pick those that are genome-wide significance, and then for each locus, we identify genes in the associated regions. That's a G, and then for each of the gene set that we are interested to look at, this could be biological function, pathway. We overlap the gene with the functional gene set, the G prime. Okay, and for each pair of genes in the G prime, we compute the shortest distance between them in the interactome. Okay, so that we are able to identify subgraph of the G prime in the interactome with the minimum distance. That's called we call it like the Steiner tree thing. Steiner tree is like an interactive acyclic graph with the minimum distance. But because not every gene would connect with each other in the interactome, so we could be dealing with more than one Steiner tree. So we call it Steiner forest, T prime. Okay. So and then after that, after we identify the T prime, we devise a statistics called S, and then which measure different components. I'm going to mention that in a minute. So then to assess the significance of S, we actually just random select loci again, and then we did the whole process to get a null distribution of S, and then we could assess the significance of S that we observe from the associated loci. So synthetic S actually have three different components. First is, of course, the number of genes in the G prime. Okay, if you have a large number of gene set overlapping genes from the associated loci, you expect that the function would, you believe that the function actually may be disassociated. We also include the number of tree in the T prime. If all those genes, these overlapping genes are connected, there's only one tree. Okay, so but if that's like all separate apart, then you have a lot of trees. So we downweight the statistic. Then we for each of the, and we have a summation to sum over all the information for each Steiner tree. Okay, and including information such as the distance in each of the trees. So if the tree is actually really large, then it means that they're actually far apart from each other. Then we downweight that. And so, right. So these are the three components that we integrate into the statistic. I just want to give you some background information about minimum spanning tree. I mean, a lot of you may be familiar with that, but MST is a spanning tree with the minimal distances across all different kinds of spanning tree. There are different kind of heuristic algorithm that we could use, primes, cross-goals algorithms. The reason that we use that is to identify these gene subgraph in the lab work. But the problem is actually not that straightforward because we are not because not every gene in a gene set is disease associated. So as an example, so if this is a gene set that we are interested to look at, okay, so every gene here is other genes in the function or pathway that we're interested, but those circles are the ones that we are that are from 4 different associated loci. Okay, so we only need to calculate the tree between these 4, but we could use information that are from genes that are outside the associated loci. Okay, so it's not only a typical minimum spanning tree problem that we face usually. So the idea is to use a COS algorithm. This is called that's like this the whole algorithm. I'm not going to go through that in detail, but one important point to note is that to calculate the Steiner tree for this 4 different nodes, we have to calculate the shortest distance, pairwise shortest distance among all these 4 different nodes, and this shortest distance could use information from genes that are outside the associated loci, okay, but in the same pathway that we are currently looking at, okay, and then so we have to keep two things in mind: the distance between them, and also keep track of what are the nodes that it passed through. Okay. The reason is that because otherwise the algorithm requires us to expand that, so we have to keep track of all those intermediate nodes. So so at the end, we are going to have a Steiner tree for that, which have all those genes from the associated loci as leaves. Okay, and then we also have information that got used from the genes from the outside of the associated loci, but within the same pathway. Okay, so as you can see, the computation of those Steiner trees actually require we keep track of all those things, and because we want to develop algorithm that could be usable and generalizable for other people that also study complex diseases. So we want to make it as computationally efficient as possible. So for MEAGA, we actually pre-compute all the shortest distance between every genes in every pathway or function that we have. Okay, so and then whenever you have some genes from the associated loci that you identify, we just look up to see what's the shortest distance between them. Okay, so we do not need to compute them on the fly. Okay, so as a preliminary look, I'm running out of time, so I just like give this a really quick overview. What's the rationale for that? This is independent of the function. Forget about the function pathway first. This is we want to see is that are those genes from the psoriasis-associated loci connected with each other, or are they closer with each other than by just randomly selected loci? Okay. So the top one is the proportion of loci pair that connected in the indactum. We're using protein-protein interaction. Okay. So this is the first thing that is actually makes sense for us to look into it. If this is not working, I mean the whole thing doesn't work, right? So so we do see that those associate proportion of loci pair connected among the top 5.5%, and when we consider the distance again, even though they are connected, they may be having a long path for them to connect. Okay, so we want to measure if they're actually really closely connected with each other. So we divided that, normalized that by the total distance, and then we see that this is like around the top 2.6. So indeed, they are both tend to be connected with each other, and at the same time, they're closer with each other. Okay, in the indactum. So we perform different simulation results. We vary different parameters such as number of genes in the G prime. We also vary one thing that's really important, actually, the distances of the Steiner trees identified from the genes in associated signals. This is a relative distance. For example, 4x means that those genes from the associated loci are 4 times closer with each other than randomly select genes. Okay, and you can imagine that 1x is actually means that those genes from the associated loci are the same distance apart when comparing with the randomly selected genes from the loci. Okay, from the randomly selected loci. Then counterexample 0.8x, which means that those randomly selected genes are actually closer than psoriasis-associated genes. Okay, but as I showed empirically, it's not the case. Okay, so we compare with the commonly used approach for the functional enrichment test in this genetic studies context, count-based approach, which is like the two by two tables that I show you. See if those function, if for each function those genes are enriched with the cis-associated loci. Okay, so there are two main observations, actually three. MEAGA tends to outperform the current count-based approach in most scenarios, especially if the interactome, the biological network, give informative information for that. Okay, which means that there's like 4x to 1.3x here, and also we actually observe that the 1x scenario is actually the same as the count-based approach. Could we expect if there's no information from the indactome, we will expect that we do not lose any power. Okay, but at the same time we remain the power. And closer, so the closer with each other, you expect that they have higher increased power to identify this associating function. And of course, if we have more genes in the G prime, we have a high statistical power to detect this associating functional pathway. Okay. So the original analysis when we did it in 2012, we used the typical count-based approach. We did not identify any functional pathway. Again, mainly because the Immunochip itself is actually enriched with genes that are in the immune-associated regions. Okay, so it's really biased. So that's why we propose to use this approach that integrate extra independent information to make the inference. So by using MEAGA, we identified 28 silicon functions. One of the really cool things that we could identify, and I'm going to show you in a minute, is that we will genes participate in the alternative signaling pathway, which have been suggested to involve in this disease mechanism of psoriasis. But now we provide a objective, a systematic way to review that. This example from a silicon function. For the starting tree for silicon function, those colored in red are actually genes from different disassociated loci. Actually, these are like 7 different genes from 7 different associated loci. And those colored in like blue is actually those genes that are involved in that silicon function, but outside the associated loci. So we do use information for that to connect a lot of those thing. Okay, so by using that, we could prioritize. Now we could give an objective way to prioritize genes from disease-associated loci, and this is a more systematic way to do that. So the next question that we want to ask is: so we now have the biological functions that we kind of have an idea, and given this objective method way to identify the approach, the function, then could we know what are the genetic variants would do on that? So we want to see if we could translate the associating signals to biological effect. What could they do? So we face different challenges. First, most signals actually do not affect protein coding sequences in a lot of complex diseases. In the case of psoriasis, less than 20% of signals would tend to affect protein coding sequence. So we need a systematic approach to identify the regulatory elements that are overlapping with the signals, and at the same time, a lot of this disease-associated variants that overlap with the regulatory elements would tend to be cell type specific. So we also need to identify the corresponding right cell type for that. Okay. So to address these issues, we perform a 5 mapping meta-analysis using the 5 different data sets that I mentioned. The 2 objectives, as I mentioned, we want to identify disease-relevant cell types. We want to identify regulatory elements among the psoriasis-associated regions. So for each of the psoriasis loci, we first perform a stepwise conditional analysis to identify if there's any secondary independent signals. Among those 41 psoriasis loci, there are around 70,000 markers that we examine, including genotype as well as well-imputed markers. And then we use linkage disequilibrium to estimate the number of independent markers, and then we use Bonforic method for multiple testing correction. So we declare the secondary signals to be significant if the p-value is less than 1 times 10 to the negative 6. Then we devise an approach to identify genomic features that got enriched among those psoriasis-associated loci, and we also include information for cell type for specificity. So first, we look at how many loci would harbor multiple independent signals. There are 10 different loci outside the MHC locus that harbor different independent signals. Okay, For the MHC locus, we have performed a file mapping studies and published in last year. So among these 10 loci outside MHC, there are in total 25 different independent signals. 4 of these 10 loci were identified previously, and 6 of those haven't been reviewed in our previous meta-analysis. So interestingly, a lot of those loci that harbor secondary signals are actually involved again in the IL-23 signaling pathway, as I show you in the MEAGA study. So and then actually like at least 5 of those actually harbor secondary signals which participate in the IL-23 signaling pathway. Just a figure shows illustrate the value for the conditional analysis. This is a this is one disease. This is one psoriasis-associated locus in the ERAP in chromosome 5. The ERAP gene would tend to like trim the peptide to be presented, so that those trimmed peptide could be presented by the HLA gene, okay, on the cell surface. So this is the meta-analysis results. We could see there's no signal, okay, and then we conditioning using the best signal imputed genotype as a covariate, and then we see the secondary signal there, okay. And actually, it seems that it turns out that these 2 signals actually the risk allele for this one tend to be on the same haplotype of the non-risk allele of this signal, okay. And because this signal is actually stronger, have a higher effect size on that, so it's masks out the signal, the secondary signal. So this is now from this example, you can see the value for doing this conditional analysis to give us a more view of how the architecture look like in the specific locus. So to give you some more information about this one, this is this is pretty interesting. It's because so again the ERAP 1 and ERAP 2 both actually would help for the peptide trimming, and in fact the ERAP 2, the best signal that I show here, is actually a cis-eQTL in skin, okay, and then the non-risk allele tend to decrease the expression of the ERAP 2, okay. The reason for that is that the non-risk allele, okay, the non-risk allele means the psoriasis decreasing allele, okay, would cause a nonsense mediate decay for the ERAP transcript, causing the low expression of the ERAP 2, okay. So and it's also been observed that the non-risk allele could actually correlate with the lower MHC class 1 expression. One of the reasons is that probably is have a nonsense mediate decay, so the ERAP 2 is not function properly anymore, okay. So they could not trim peptide correctly, so that the MHC actually could not present the trimmed peptide well, okay. So and this is how we could visualize how this ERAP 2 could have the ERAP 2 signal could have protective effect for the psoriasis. That again the no risk allele lower the ERAP 2 expression by causing the nonsense mediate decay, and then causing lower MHC class 1 level on the cell surface. And because this is autoimmune disease, so lower for that, lower MHC class 1 may cause low risk of psoriasis. Okay, so this work is really nice from a post-genetic publication. So then the next question we want to ask is whether the secondary signals could help to increase the variance explained. So altogether, among all the non-MHC loci, the secondary signals would contribute to around 20% in variance explained. And altogether, including MHC, because MHC actually have multiple secondary independent signals for increasing the risk of psoriasis. Altogether, the secondary signals could help to explain the variance by 40%. So we implement an approach to identify genomic feature that got enriched among the independent psoriasis signals. We calculate this enrichment among 9 different chromosome states that common in 8 different ENCODE cell types. Those blue color representing those are significant. So among all these different X cell types here, the most significant one are the keratinocyte. And keratinocytes consists of over 90% of the epidermal layers. And then we also see the lymphoblastoid cell line being the most significant one. So among those different chromatin marks that we look at, the H3K27 acetylation one is the most, which indicated active enhancer is the most significant one. So this actually motivates us to look into those marks in different immune cells. And I'm going to show you that in. So okay, before that, because LCL is was one of the most studied cell lines in ANCO, so we also look into other different genomic features to see what others are enriched. These are the top 7 features. Besides those histone modifications, we also look at different transcription factor binding site, and we observe some of those transcription factors that are involved in the immune mechanisms, actually, like NF-kappa B, LF4, actually, the binding site are actually enriched as well. Okay. So again, because the H3K27 acetylation was the most enriched one in LCL, so and then we also want to see that we also want to see if these are enriched among different immune cells. Okay, so we use those analysis predict in a recent study and perform enrichment analysis, see if there's enriched among those psoriasis-associated loci. Those in brackets are those the number of secondary signals that overlapping with the enhancers in corresponding sites. Okay, so we can say the most significant cell types are the T cells. Okay, and then another observation is that besides the primary signals, we also see secondary signals being overlapped to have the regulatory roles by predicted by the enhancers in corresponding cell types. Okay, so I'm going to show you a couple more slides that how we apply the MEAGA as well as function enrichment and genomic feature enrichment in our most recent analysis. So this is so all those things that I mentioned to you are based on genetic association studies that consist of over half of those are Immunochip samples. Okay, so and in fact, more than half of those psoriasis loci identified in Caucasian were identified based on Immunochip-based studies. It actually limits the fact that we are able to identify loci outside regions from the Immunochip. So to address this, we coordinate with other different groups, and we gather different GWAS data sets, and also including our page Immunochip data sets. This 7 GWAS group consists of those that have been published, and also results from 23andMe. Okay, so we are able to the effective sample size for this whole samples, whole data sets are actually over 30,000 genotype individuals, only for the GWAS component alone. Okay, some of the highlights include those we found systematic difference between clinically diagnosed versus self-reporting cases. We also are able to increase the number of European origin psoriasis loci to 63. But actually, I'm going to show you. I'm going to relate that to the things that I was talking about earlier. So, okay, before that, we show that there's like a as shown in other complex diseases, there's a correlation between the effective sample size, effective size for the disease-associated loci versus the modality frequency spectrum. Now with this more than 60 psoriasis loci, we are able to have pretty good area under our OC. And at the same time, when using the effect size of different loci as to devise a genetic risk score, we are able to correlate that with the age of onset of psoriasis. Okay, so those people with so we separate that because MHC actually again has a largest effect size. on the risk of psoriasis, and we implement. I mean, we compare the model with or without MHC. Both of those are see correlation between the age of onset of psoriasis versus genetic risk score. For example, for those with the high 5% of genetic risk score, they have to have an average age of onset of around 20. But for those that with the lowest genetic risk score, they have an age of onset of around 35. Okay, which is pretty significant difference. So we applied MEAGA on the all the disease or the psoriasis associated loci genes, and in addition to the alternate free signal pathway, we also highlight pathways such as alpha kappa B. Okay, alpha kappa B is actually one of the downstream signals for alternate free signal. So now we have a more complete picture for disease mechanism, and the genomic feature enrichment analysis is actually just concordant with what we observed before. You see the best. We see those T cells with the enhance of T cells actually highly enriched among all the disease psoriasis-associated loci.
Yes. So in cirrhosis, mainly three things happen. First, your leukocyte becomes stronger, right? You have more interleukin. Secondly, your cells respond more strongly to immunosignals. For example, your NF-kappa Bs are proliferated. Third, some leukocytes are not in the right place. They might be in normal tissue. In normal human, they are in the blood, but in cirrhosis, they might in the tissue. For misplaced for leukocytes, they are not placed now in the right location. Do you find any genes that are related to this process?
Okay, I'm sorry. You mean for the nucleus?
For infiltration of
immune cells?
Yes. So do you find any genes related to this process?
Oh, you mean do I find any genes that involve in the infiltration?
Yes.
Oh, okay. So yeah, that's a good question. So I think it's I'm not the best person to answer that, but it's something like that. So we also see a lot of people that first developed some lesions there, or they got hurt on the skin, and that would trigger the psoriasis. Okay, so that actually an environmental factor involved in that. So the T cell here actually is actually a good point that so these are T cells, okay, but they may not have a skin homing component. This is actually what we are observing. We are now looking into. We are using ATAC-seq here to try to identify regulatory elements for the skin homing T cells. Okay, so, and then we are going to identify the enhancers, potential enhancers among those skin homing T cells to see if they're enriched among genes from the disease from the psoriasis-associated loci. Yeah, but yeah. So I guess the T cells enhancers indeed are enriched among those psoriasis-associated loci. So which is actually we have to find what are the genes that are regulated by the enhancer. That's the next question, right?
So why your MEAGA cannot find the infiltration-related genes until now?
So when you say infiltration-related genes,
so genes that make those T cells not staying in the blood but get into the tissue.
Oh, okay. So these are the so for example these are the genes. Okay, this is actually going to the keratinocyte. Okay, so this is causing the infiltration because there's like so. I guess I'm not sure if these are the direct drivers for the infiltration, but at least these are some of the process that would causing the that involving the alternate free signaling pathways that are causing this kind of abnormality in the skin layers. Okay, but I'm not sure if these are the main drivers for the infiltration. Okay, all right. Okay, so as I mentioned, so we are trying to understand the regulatory effects for the associated signals, and we are trying to see if we could also characterize the chromatin landscape for skin homing immune cells. We also want to do chrome QTL, try to associate the genomic variance with different chromatin states. And for the bioinformatics part, we also try to extend the current system biology framework to include other different independent data. So we've been trying to identify this potential disease-associated cell, trying to identify polarized candidate genes with the associated loci. We could think about a way to try to integrate different kinds of information. Okay, and besides the we've been only using the protein interaction, we could also look at to see other biological network that we could use to integrate another layer of information. Okay, so so this is like highly collaborative work, including all of my colleagues in here, and also collaborators from UK and Germany, and also a lot of collaborators from the two consortium contributing to Immunochip data sets. And I especially have to thank JT Alder and Gonzalo for the guidance and also for the help during the whole project. All right, thanks. Any questions? All right, thanks.